Hello. So it's time to start again for the afternoon session. So we continue the action session with presentation from different working groups. And as this morning, several people were involved in the preparation of the presentation. So this group is about human microbiome and public health targets. It involved Jeroen Raes from VIB-KU Leuven, where he's professor. Professor Robert BENAMOUZIG from Assistance Publique Hôpitaux de Paris, and Matt Henn. I don't know if is Matt online? Yeah. Oh, so Matt is online. SeresTherapeutics, and Jeroen will present for the group the presentation. Please join.
Okay, this is the clicker. All right. So I learned from Per that I have to speak in this thing. Good afternoon, everybody. After the wonderful lunch and the wonderful lunch temperatures, I have the honor to kick this session on the human microbiome off. So this is an action session, right? So there's no pretty pictures in this talk. There's only things that we think that should be done. And this is, I would like to emphasize, this is also an invitation. This is not, we don't want to dominate and decide what needs to be done. We have some ideas, some proposals at various levels. Some are more towards policy, some are more towards science. But I would like to stress that we are also looking for input from everybody to make this working group a success. So when you think about the role of the microbiome in public health, there's many things you can think about, and this is a slide that actually came about with input from Joël as well, where we think, okay, how can our working group or how can the WMP help impact public health? And I think some ideas are that we can use it to help control the rise in incidence of chronic disease, given the implication of the microbiome, in this case the human microbiome in chronic disease. I think we will also be able to assist in maintenance of health, prevention, preventative science. I think it will help us. The WMP will help us promote the clinical benefits of microbiome monitoring and modulation. It helps standardizing the diagnostics. It will help us spreading the microbiome monitoring to previously less covered areas. This has been said a few times across multiple areas of research, but this is also true for the human microbiome stuff. There is a lot happening worldwide, but there's still not enough happening worldwide, and many areas are still very much understudied and undersampled, and we should not forget those populations as well. And the link between diet and the food chain, as we discussed before, I think is going to be very interesting as well. So after some discussion between the different members of the group, we thought, okay, what can we do and what can't we do? At the moment we are budgetless, so what can we do? And I think we focused on 3 different areas which we would like to explore. And so the first area is the area of the reference cohorts of healthy populations. Right? So this is human again, healthy human reference cohorts to better enable microbiome research, because they act as a reference to all the clinical studies and serve as a backdrop for all kinds of applications. The second focus area we focused on was the development of the support of microbiome diagnostics. There's a lot of talk about microbiome diagnostics. There's spin-off companies arising, but still the first real clinical microbiome tests are not out there yet, and there's still some work that needs to be done there. Then the third area is where was in recommending priorities for all of the stakeholders on the modulation, right? So the application technologies for health maintenance, disease prevention, and cure. So why focus on reference cohorts? And I think this is interesting because the question has fallen 2 times already this morning, so it's clear that this is one big elephant in the room still. We don't know what a healthy microbiome looks like, right? It is very, very difficult to understand where the borders between healthy and what we then in the human-centric microbiota world call dysbiotic. We start to understand, we know some properties of a dysbiotic microbiota. So everybody can name some features, low diversity or low load or more less butyrate producers or whatever. Everybody can imagine something, but where's that border? Where does that stop? When do you tilt, right? And it's important because in terms of reference ranges, in terms of diagnostics, you need to find where is that tilting point. Where do you say bad? Where do you say good, right? And the problem is that even in the healthy microbiome, even if we can say what is dysbiotic, the healthy is also very difficult to really define because there's a lot of variation. And we don't always know what that variation means. There's a lot of contradictory results. For example, people that are Prevotella dominant in their guts, that has been associated with, let's say, a more fiber-rich diet, and it has been associated with rheumatoid arthritis, right? So it is very, very contradictory results, and we still don't really know what to do with that. And the question is there one healthy microbiome, or are there multiple healthy microbiomes? Is there one dysbiotic microbiome, or are there multiple dysbiotic microbiomes? We don't really know. We also don't know the long-term outcomes. If I today have a dysbiotic microbiota, what does that mean for my long-term health? We don't know these things, right? And then definitions matter. I was part of a discussion group some years ago on a healthy microbiome, and I think the first half day of that session, we discussed about, but what are we really saying? Are we saying, is a healthy microbiota the microbiota of a healthy host, or is the healthy microbiota the microbiota of which is consisting of a healthy, stable ecosystem, right? It's not always the same, right? And so lots of different questions. And I think this is where reference cohorts of healthy individuals then can play a role and have played a role. There's been plenty of papers that have been triggering this already. So the current state is that there was pioneering work by the first studies in the field. I'm now focusing mostly on the human microbiome and the gut microbiome. So things like the European MetaHit, the American HMP, they really spearheaded everything. And then you had a bunch of population cohort studies that had the potential of acting as reference cohorts for the healthy population. And so I named a few here: French gut, American gut, Flemish gut, Twins UK, the HPFS. There's plenty others, right? This is not an exclusive list. I just took 4 or 5 that came up, but I know there's plenty out there, right? And so all of these different population cohorts, they're doing a really good job at sampling their populations with a lot of metadata, with a lot of omics data collected, but at this moment they don't talk to each other, right? And I think that's where the future, possible future, should lie. They sometimes replicate on top of each other. We, for example, work together with Lifelines and various other combinations, but they are, at the moment, they're somewhat isolated. The current state is also that the study is primarily focused on taxonomy, mostly, and now the taxonomy is all turned upside down, so then we're in deep trouble. And also the biggest problem is also that the way samples were collected and the data was collected and the samples were treated and the DNA was extracted is very heterogeneous. I think it was Per who said it, or Jeroen who said it. There are standards, but people are not using them, right? So then if you don't use the standards, then, well, it becomes very hard to compare. There's also differences in what metadata and other data is collected on these individuals. There's differences in the design. Some studies are purely cross-sectional. Some are panel studies. Some have longitudinal arms. That is not homogeneous at this moment. It doesn't have to be, but that's the state of the art. And so the opportunities for that we as a working group defined is to, first of all, do a mapping. Map where the gaps are. What are we not covering? Hey, I already mentioned geography. I think that we urgently need large population cohorts in South America, in large parts of Africa or Southeast Asia. There's things happening, but I think we need a lot more. And the things that are happening there, I think we need to reach out. We need to work together. We can exchange protocols and things like that to make that happen faster. I think we need to establish some priorities, right? What are we going to do if we want to collaborate? What things will we be focusing on? What extra data will we collect? What type of profiling will we do, et cetera, et cetera. And then this is what Joel already alluded to. We would like to establish working title, the Microbiome Observatory for Human Health. Right? So what it means is it's a connected network of population cohorts who talk to each other, who exchange information, who exchange to try to harmonize protocols as much as possible, so that we can actually start looking across these different large-scale population cohorts to understand the variation. And then from that work, I think we can also recommend some best practices to improve the alignment of such protocols. This is really a problem. I was at one point involved in a GWAS study where we tried to find loci in the human genome that explained the variation of the microbiome. It was a huge meta-analysis across 20 different cohorts worldwide, and the end result was actually very disappointing. And the reason why it was disappointing was because the data was so noisy, the protocols were not aligned, and we could not just find the signal beyond the noise, and that is really problematic. Okay, on the diagnostics arm, this was the second point. So the current state is that long-term dynamic monitoring has not been done much. So how can you do diagnostics if you don't know the dynamics, right? There is some potential in colorectal cancer, inflammatory diseases. Biomarkers have been proposed. Some companies have been started, etc. So I think there is a potential there, but I think we need to get more of a consensus on what are really realistic targets for diagnostics, and how that we're going to do this, right? And I think there is not enough alignment yet, or maybe we're still early in the game. And so we also need a framework to regulate microbiome testing. There's tons of companies out there that provide microbiome testing at very poor quality, to be honest, and in terms of reporting, they do horrible things. I think there needs to be some regulation there as well. There was a recent initiative from Ianiro in Rome, who basically, in a big working group, decided that most of what is out there is actually not high quality, and formulated some recommendations. So it's also unclear at the moment what technologies are best used. Do you use PCR-based single markers? Do you do whole sequencing? Do you do single point measurements, multiple point time measurements? We don't really know, and still we want to be doing diagnostics based on the microbiome. So there's work that needs to be done. So at the moment we decided to create as an action point a panel, an advisory panel, a subgroup, to discuss all this, and everybody who is involved or invited in these types of things is obviously welcome to join this, so that we can start working on what data types, what dynamics give the greatest near-term value for doing these diagnostics. Is it just health versus disease? Is it dysbiotic versus not? There's all kinds of distinct things you can do, but we also need to decide how we would be doing this and what we as scientists would recommend to do, and prioritize some use cases for the best type of diagnostics. And then the third action is the area that sort of overlaps with the treatment arm, the other working group, where we as a in the health perspective, we would like to also develop a group that helps prioritize the disease areas that people can focus on. And there's a lot, again, we're in an area where there's a lot of promise, but at the same time, there is still a lot of disease areas where we're realizing that maybe the microbiome application is going to be more difficult than in other areas. And I think it's time to sort of start to focus a little bit, I think. So there's the area of biotherapeutics is something that is growing. There are 2 products on the market now in the US, not yet in the EU, but that's it at the moment. And all the others are still in the pipelines. On the academic side of things, there's a lot of activity in the FMT space, fecal microbiota transplantation. In different areas, you see them there, but these have not translated to the clinic yet. And still, I mean, although this is all very promising, the cost to really push this from, some interesting new potential findings and interesting to really treatments and medication, getting them on the market are enormous. And I think it needs to be some more coordination is necessary. And this is also a demand from the spin-off community. They would prefer that there's a more common push towards this area. And so again, working group at the moment to make it more precise, but to define the near-term prevention and therapeutic opportunities and the gaps, and maybe work together to synergize on the investments in this area, because taking microbiome science from a microbiological science into a real pharmaceutical clinical discipline, it costs enormous amounts of money, and I think it's better to cooperate on that front. So to make it concrete, we had some first ideas. You are willing to, you are completely able to allowed to disagree with these ideas or agree with these ideas. One of the ideas was that we do an analysis of these different key themes. We write a white paper around that. Again, this is something that can be done and will be done in community sense, so everybody who's interested to contribute can contribute. Something we discussed also, given, for example, this morning, all the big ideas on policy and One Health and things like that, there's still a big discrepancy between the big policy ideas and then the hands-on science in our case, in the clinic. And so I think what this initiative can also do is allow for better communication between policymakers and the scientists to link these big ideas and to maybe to test the feasibility of these big ideas in the reality of science. So we would like to establish an advisory board for gap finding and coordination of these reference cohorts. We would like to transfer that into a microbiome observatory for human health, and then build a strategy to fill all of the gaps that we want to, that we're seeing in this area. And so I'm aware that these things are somewhat concrete and they could be more concrete, right? This is just first ideas. I think this is something that we, I think, should be doing together with the people that are. So, oh, sorry. So these are, so the last slide was saying, could you just get me the last slide? I'm sorry. So these were some first ideas that came out of the discussion between the three of us, but the goal is actually that this is an interactive session and that we can get some feedback from you guys as well. So if you have further ideas of what you think is important in the microbiome public health space, this is your moment. Thank you.
Thank you, Jeroen, and thank you to the three.
Hi, Matt. Good morning.
Contributor. Hi. Is there questions?
Yes. Just one potential space also is all the things related of the interaction between drugs and gut microbiota. Because then you can start with existing data cohorts. There are data, for example, from statins and gut microbiota to predict the response, and this could be maybe a way to leverage the existing data set for existing drugs. This could be one way. And the second point, I don't know whether we can also look at the small intestine, because this is the missing piece as of today, especially if you think about the interaction with any drugs or treatments, and also the vaginal ones. But there is a nice ongoing initiative by Sarah Lieber. So I think this could be some interesting thoughts.
So I agree completely. The drug microbiome interaction is a very nice opportunity, for example, for repurposing of existing drugs, so we can very quickly go to new treatments. You've seen the panel composition. We have a bias towards the gut microbiota. I think we warmly embrace the other microbiotas of the human body into our working group. So the vaginal is one of them, and skin, and I mean, they're all equally important, I'd say. So these are good ideas. About the small intestine, I think it's also a very important area. It's been plagued by a lack of accessibility. It's more invasive sampling. But yes, I agree completely that this is also a focus area. Yeah. Karin?
Yes, just to answer a quick note, because in the introduction from the president of INSERM and in Raes this morning, so they mentioned the French, which is called PER, which is large-scale study on the gut microbiome. So one of the projects, actually at least two projects on small intestine has been funded, one on the domain of metabolic disease and another one on inflammatory intestinal disease. So, and well, one of them I'm coordinating one of them, but yeah, we hope we can provide in the future more information in this intestine region.
Hi, thank you. I don't think we're going to get very far with reference cohorts. I mean, we've been at this for 15 years now. That's Matt Henn, right? So, hi, Matt, Jim Versalovic. I mean, Ceres is an example of success with defining a microbial consortium and actually treating disease without knowing that much about microbial composition. So the point is, in human medicine, we only have one example where we look at differences in cell populations, the cell differential, complete blood count, and it's limited. Really, it's biomarkers that are functional biomarkers and biochemistry, cholesterol, LDL, glucose. That's really what we need for the microbiome, is to define those functional markers for health and disease, and I think there the effort should be on function, not composition.
So let me get one thing straight. The cohorts, the reference cohorts, are a vehicle. I was not focusing on the taxonomic output only, right? These cohorts, what I mean is that they are sample collections often. Most of these cohorts have thousands and thousands of samples. Many of them have multiple samplings over time of individuals. So if you and I think we put it in the slides somewhere, the functional readouts are important. And with functional, I don't really only mean metagenomics, right? It's also finding specific peptides, specific metabolites, and things like that. So I think we actually agree. Okay.
Yes.
Thank you so much for this nice communication, and thanks for all the present here who I consider as pioneer in this world of microbiome. I'm an oncologist. I'm fighting cancer since 25 years now, and I got infected by this microbiome virus a few years ago. And I just wanted to share the perspective of oncology, right? We made some progress over time, over the last 30 years. And these ones were incremental, right? So when we try to look at the cell biology and the cancer biology and try to identify and map all the paths and try to have a full picture, like at one single point, a snapshot of everything so we can be in full control, we lost time. When we started learning and implementing from the learning that we know, step by step, we started putting targeted therapies, immunotherapies, and we started moving the needle forward. So I just wanted to say that maybe, due to this complexity of this word, like the trillions of interactions and the interactions of the microbiome itself with the other entities, whether it is the soil or our own cells, maybe validate small wins each time, in order to move forward and not wait until we have the full picture on everything. So we can, we knew, and like in infectious diseases, that some bacteria, you need to kill them because they kill you, right? Maybe having like a similar approach to where once we validate something, we know, we identify pathobions, new ones that were not visible to us like 30 years ago, let's try to go after them, or and so on. So that's a comment, a general comment, just maybe to bring something to the puzzle.
Yeah, I obviously agree with you, but I would also say that is happening, right? This is what we presented here was more about global cooperation and things like that. I think the small wins are being pursued at this moment. Whenever an interesting target pops up, for example, in the colon cancer world, there's a whole horde of people going after fuso. People are picking up on interesting targets and following them through. Ceres, as an example, and of all the biotechs out there, are doing exactly that. They're not swimming wildly. They're going after very, very specific targets. Matt, maybe you want to comment on that question as well. Did you hear?
Yeah. Can you hear me? Can you guys hear me?
It's okay, but the quality is not so fantastic.
Okay. Yeah. Well, good afternoon, everyone. And sorry, it looks like I'm a major face looming over Jeroen, so hopefully I'm not scaring you too much on the screen. Yeah. So I would say that I want to comment on Jim's comment as well, because I think it's spot on. And it's certainly how we're thinking about it. And I think it's connected to the question that was just asked. As we think about developing drugs and new technologies, as we've talked about as a group, and I'm sure you'll hear in the next working group, it's really, really important to be able to think about function, what you're specifically targeting, and most importantly, what is the heterogeneity across the patient population that you're trying to target, such that you can actually ultimately design a clinical trial to demonstrate effect. And I think our view as a working group, and welcome others' opinions, is that we have an opportunity to try to aggregate across many different efforts to try to pull out those learnings and apply them in the context of advancing the space. So I think these are all really, really important points.
You can repeat if you catch it.
I think, Matt, the summary of what you said, because the quality of the sound is very bad here, so we have a hard time understanding. But I've understood that you said that focusing on function is probably very important. I think that's something that we've emphasized as well, and that the aggregation of all the data in the different cohorts is relevant also for companies like you. Is that correct? Yes, that's a yes.
Very good, thank you. So thanks a lot, Jeroen and other contributors.